Welcome to an hour of HealthMade Radio. HealthMade is a community for natural health seekers where we educate people about common health conditions and share extensive research on the most effective natural health treatments and promote legislation that protects our health freedoms. A core concept belief is in the innate intelligence and healing power of the body, and if properly supported spiritually, emotionally, and nutritionally, it can find its way back to health. HealthMade Radio will bring you information from integrative health experts throughout the world. Check us out at healthmade.co. Health is what you make it. I'm Dr. Michael Karlfeldt, and I will be your host. Today's guest is Dr. Don Huber. Dr. Huber is a professor emeritus of plant pathology at Purdue University. He was serial pathologist at the University of Idaho for 8 years before joining the Department of Botany and Plant Pathology at Purdue University in 1971. He retired in 1995 as Associate Director of the Armed Forces Medical Intelligence Center as a colonel after 41 years of active and reserve military service. His agriculture research the past 50 years has focused on the epidemiology and control of soil-borne plant pathogens with emphasis on microbial ecology, cultural and biological controls, and physiology of host-parasite relationships. Research also includes nitrogen metabolism, micronutrient physiology, inhibition of nitrification, and nutrient-disease interactions. He's received various awards for scientific accomplishments and contributions to government. Dr. Huber is an active scientific reviewer, international research cooperator with projects in Argentina, Australia, Brazil, Chile, China, Costa Rica, Denmark, Germany, Japan, Mexico, and Russia, and a consultant to academia, industry, and government. He's the author or co-author of over 300 journal articles, experiment station bulletins, research presentations, book chapters, and review articles, 3 books, and 84 special invited publications. He's internationally recognized for his expertise in the development of nitrification inhibitors to improve the efficacy of N fertilizers, interactions of the form of nitrogen, manganese, and other nutrients in disease, herbicide nutrient disease interactions, techniques for rapid microbial amplification, and cultural control of plant diseases. He is an expert when it comes to the topic of what we're going to talk about today. Dr. Huber, it is such an honor to have you here on the show. First, we're going to be discussing GMO. And what made you interested in that subject?
Initially, Dr. Karlfeldt, I looked at the potential of GMOs, and I was very excited as a scientist to think that we could do some little bit of tinkering here and there and make some great strides as far as breeding programs and that.
So this was actually a project of yours that you moved forward in regards to GMO.
Yes, I actually had the first permission for field testing a GMO in the state of Indiana.
That's fascinating.
So we were modifying a bacterium that characteristically fixes nitrogen, and we found that if we did a little modification from another bacteria, that we could actually get 4 times the nitrogen fixed from the atmosphere that we could without that.
So what kind of issues did you feel that you were going to solve with GMO? I mean, why do we want GMO according to what you thought at that time?
Well, our concept of what we were doing with GMO, of course, at that time, that's 50 years ago, today it's fossil science. We abandoned that whole concept of one gene, one function, literally 50 years ago with the sequencing of the human genome. We found that it's not quite that simple, that our understanding of genetics today is really a concept of a spatial relationship between the different genetic materials in the cell, and the influence then of environment as it works on those different genetic materials, so that you may have one thing that's happening that you're looking at, but there are another 40 to 200 new proteins that are also being produced that we have no idea what their safety is, or toxicity, or even their function.
So with the GMO, it's not you test it in a controlled environment, but then when you do these genetic manipulations, and there's a different environment, or the environment changes, you really have no idea what the end product is going to be.
Correct. We have just a few more genes than an earthworm. Now, we had anticipated 50 years ago that we would have over 100,000, because we know there are that many functions going on in the body. And if it was one gene, one function, we would have a gene for each one of those functions. Now, with only 19,000 genes, we know that each gene can perform many functions, or direct many functions. And many of those functions we're still in a primitive state of knowledge on. When it comes to genetic engineering, then, when you change that spatial relationship, and it doesn't matter whether you're inserting or whether you're deleting or silencing, as we call it, editing now.
So what does, when you say spatial relationship, what does that mean?
That's the interaction of one genetic piece of material with an adjacent piece of material. Now, we picture the helix and everything in a two-dimensional form, but that's not the way it really exists in the cell. It's more like a Rubik's cube, so that when you change one thing, you're really changing literally hundreds of other things that are influenced by that genetic material. And then as the environment changes, that effect is also multiplied many times.
So is there a way to predict as to what kind of changes that you create by inserting a genetic material?
We can look for specific traits, but we have no idea of even knowing many of the other traits that are being changed at the same time, because we don't even know what they are. One example would be with the genetic engineering of the bacterium to produce a very essential and necessary amino acid, L-tryptophan, important for regulating or controlling many diseases. But when they genetically engineered that organism, there was a new product that was produced that took us 12 years to identify. The only reason they persisted for the 12 years was that within 2 weeks after introducing that product, the genetically engineered product on the market, it was so acutely toxic that 80 people were dead, and 10,000 people now are permanently incapacitated from the anaphylactic shock that it caused.
And that was due to the GMO that they did at that time, the bacterium.
Right.
Yes.
In addition to the L-tryptophan that it was producing more efficiently, it also produced 3-PAA, phenylalanine amide, and in that process, that product we had never seen before, wasn't in any databases, and it literally took 12 years of some of the best science and techniques that we had available to even identify it, to find out what it was. So when you genetically engineer a plant just for the Roundup Ready trait, for instance, we know that there are probably at least 40 new proteins besides that EPSPS protein that gives it the Roundup Ready characteristic.
Yeah.
No one's ever tested any of those products for safety. If they're chronically toxic, in other words, if they build up over time in the body, which many things do, that it's very difficult to identify a cause-effect relationship because it's building up over time. It may take 20 years or 30 years. You look at the tobacco cancer interaction, we worked 20, 25, 30 years before that could really be established strong enough that people were willing to accept it.
So when they were introducing then this genetic material, and they use like a vector, like a bacterium or a virus or so forth, to introduce this genetic material into the genome, that then, and their only desire is then to change one aspect so that it's able to withstand more pesticides, so it can handle more pesticides, and that way they can spray more and kill more weeds, and still what we're supposed to eat will survive all those pesticides. But when they look just for that one change in trait, but you're telling me that there can be 40 to 200 other proteins or substances that are created from that really we have no idea what they're going to do. And just like when you do drug interactions, if you have one drug and you take another drug, that becomes what's a drug interaction between those two. And then if you multiply that 40 times, you really have no idea what kind of effect you're going to get over time. Right.
Correct. That's correct. In fact, the EPA just announced last week that they weren't going to regulate gene silencing, the CRISPR techniques. And yet today we had two very excellent scientific papers that were published. One of them by Novartis, which is has a pretty good investment in genetic engineering, but they felt that the data was significant enough that they permitted their scientists to go ahead and publish it because of the potential damage that could do. The other paper on the same general type of research was from the Swedish Medical Institute, showing the same thing, and that's that one of the unintended consequences of silencing a gene is that it also silences another gene that's called the P53. The P53 is what keeps you from getting cancer. And so when they silence that gene, what they're finding in the mice is that you essentially guarantee that an individual will develop cancer, even though there may be some other positive aspects. You're just choosing a different poison in the process, one that may take a little longer to kill you than, say, muscular dystrophy or Parkinson's disease, but it will occur.
Yeah.
And so there is so much that we don't know, and that became very clear to me as we were looking at some of these techniques, that there may be great potential in the future when we understand how to do it and what we're doing. Currently, we have no idea what the consequences will be.
And when we will be right back with Dr. Don Huber, talking a little bit more about the unintended consequences in regards to GMO foods that we're now experiencing. We're back here with HealthMade Radio. I'm chatting with Dr. Don Huber. He's probably one of the leading experts in regards to GMO and GMO technology. So you were talking a little bit about some of the unintended consequences and in regards to GMO and the silencing of a gene technology. And one of them then being cancer. And we're experiencing a huge rise in cancer, where almost 1 out of 2 will either get cancer or 1 out of 4 will die from cancer. So do you feel that there is a direct correlation with GMO?
I don't think there's any question. Scientifically, that's been very well established, that there is a direct correlation.
So with cancer, because it's so hard to think, here you're eating a carrot or you're eating a strawberry and thinking that that is something that is going to cause cancer, and then genetically modified organisms and actually drives that process.
Well, there are two factors that are involved. But first, there are two very excellent studies, lifetime studies, and with rats that document the carcinogenicity of both the GMO process as well as the products, the glyphosate product, that 85% of those plants are genetically engineered to tolerate. Now, one of the factors just in the GMO process that the Seralini study showed was that the cancers come on much earlier and much more intense. The 2-year study that Monsanto conducted that has just been released as a result of the non-Hodgkin's lymphoma lawsuit in California shows that actually there are 12 or 13 different cancers that are all increased when you expose the animal to the glyphosate, and the same thing with the GMO foods. Dr. Shiva Ayyadurai and his team at Massachusetts Institute of Technology 2 years ago published 4 papers showing what that process was, or connecting the dots for the process for carcinogenicity from the GMO process and those unintended consequences. One of those processes that happens when you insert new genetic material, or when you upset that spatial relationship, is that you block C1 metabolism. C1 metabolism is how the body gets rid of all of your trash materials or your carbon compounds.
So all the metabolic waste, as you know,
and that process, what you end up with is formaldehyde. Well, that formaldehyde would normally be very rapidly picked up and metabolized to CO2 and water, so that it's not a problem for us. When you disrupt the integrity of the genetic code, though, then you have formaldehyde accumulating in the body. Formaldehyde is a Category 1 carcinogen. It explains why we see that increase in a lot of these cancers. The other thing that happens that Dr. Ayyadurai team showed was that when you have an accumulation of formaldehyde, you also have a complete depletion of glutathione. And in spite of having 3 or 4 different ways that you could normally jumpstart your glutathione, adding cysteine and increasing that in the diet, several other ways to do that normally, but when you have disrupted the integrity of the genetic code with C1 metabolism block, none of those processes work. So that you see the increase in cancer then because of the carcinogenicity of products that are produced from disrupting the genetic code. The other thing is when you're adding the glyphosate or the Roundup, which 85% of our crops are engineered to tolerate, that there's a direct carcinogen that you're adding, and that's N-nitrosamine. Now, that's the tobacco carcinogen. And you'll find that every bottle of glyphosate contains N-nitrosamine as a contaminant, just like the dioxins were for the phenoxy herbicides for Agent Orange.
Yes.
So we're not only changing the metabolism of our body or our GI tract organisms and the plant's metabolism, but then we're also consuming carcinogens that we used to not be exposed to at all.
So we are silencing genes that then also interfere with substances in our bodies that helps us fight cancer. And then we're adding carcinogens into the food, like the nitrosamines. And also, obviously, the glyphosates in themselves, I would assume, will have a toxic impact in large enough quantities. And we're increasing then formaldehyde in our bodies because we can't get rid of it. And then also we are then reducing our ability to produce glutathione, which is one of our main antioxidants and is also crucial for energy production within the cell, which will shut down the mitochondria and our ability to move heavy metals and chemicals out of the cell. Also shut down our liver function if we don't have enough glutathione, and they see a direct correlation that the less glutathione you have, the shorter you live. So all of that takes place with GMO foods.
Right. With such a quote-unquote simple process, genetic engineering is more like a virus infection than it is a breeding program. And a virus does the same thing. It inserts or it silences, but it changes that spatial relationship. And when you do that, then you have all of these other unintended consequences that science has never seen. We've never studied them. We've never looked at them. But all you have to do now is look at the Center for Disease Control statistics on the occurrence and increase in about 32 diseases that can all be traced back to these functions that we do know are disrupted by the genetic engineering process.
And this is just a tiny piece of what we know, correct? So when we come back, we'll talk a little bit more about the impact of GMO with Dr. Don Huber. You're listening to HealthMade Radio. I'm here chatting with Dr. Don Huber, one of the leading experts in regards to GMO technology and the impact of GMO in our food supply. So you were mentioning that when this started about 50 years ago, that it was based upon, it was called ancient technology or ancient scientific understanding. And now we feel that we have a much better understanding, even though we're using the technology that we had 50 years ago. And still, obviously, as technology advances, then we will understand more and more the impact of GMO foods on our physical bodies.
Correct. We're still basing the whole concept of genetic engineering on that fossil science that was proven wrong 50 years ago. And that's one gene, one function type of a phenomena. So that as long as that's the predominant dogma that is used in the process, no one looks for all of the other unintended consequences that we know can be extremely fatal from the L-tryptophan experience. We know that from the Pustay research with the genetic engineered potatoes that was stopped very abruptly as soon as he started seeing the potential for lethality from that, so that we have no idea what the cause is on many of these things. We just see the disease pressure increasing and keep our head in the sand and say, no, it's one gene, one function.
So, yeah, we're seeing more, like we're talking about, more cancer, more chronic diseases, more neurological diseases, people dealing with Parkinson's, ALS, MS, seeing a lot of the autoimmune conditions, like digestive, dealing with Crohn's, colitis, ulcerative colitis, seeing also issues in regards to muscle wasting, cognitive, more anxiety, more depression, dealing with more diabetes, obesity. So all of these things, in essence, can be pointed towards that GMO is a strong factor in.
Well, you have the GMO, but you also have the other factor, and that's the chemicals that it's engineered to tolerate.
Yes.
And so with 85% of them tolerating Roundup or glyphosate, that chemical alone is a very toxic chemical. It just doesn't have the acute toxicity like the 3-PAA with the L-tryptophan, but it's probably the most chronically toxic chemical we've ever released into the environment. And that's because it's a very powerful mineral chelator. so that it ties up your iron, your manganese, your copper, your cobalt, zinc, all of those very critical amino acids or minerals that function as cofactors for your enzymes. All of those are immobilized by glyphosate. The other thing is that it's a very powerful patented antibiotic. But it's an antibiotic against the beneficial organisms in our GI tract and in the soil and that. But it stimulates the pathogens so that Clostridium, E. coli, Salmonella, Listeria, all of those organisms are stimulated in their virulence and their disease-causing potential, while all of those organisms in our GI tract that would normally suppress those pathogens are all very sensitive to the glyphosate and removed. So that it's very rare to find a void in nature. When you take out one group of organisms, and in this situation with glyphosate, we're taking out entire groups of organisms, then it leaves the disease organisms then to fill that void, and they do that very well. So that all of those diseases that you mentioned, from autism, Parkinson's, bipolar, all the muscle wasting, the autoimmune diseases, all of those have a tie directly back to the GI tract. And it's that group of organisms in our GI tract that produce the neurochemistry, the aromatic amino acids, all of those compounds that we rely on for survival. And without those beneficial organisms, then we see all of the deficiencies and the disease developing as a result of that.
Yeah, because you have in the gut, you have about 70% of your immune system is along the gut, and you produce at least 30 plus different neurotransmitters. Neurotransmitters are the hormones that regulates how we feel emotionally, and also it signals any kind of signal from one part to another part of the body as to what action should take place. Like, for instance, if your muscle should contract, there needs to be a neurotransmitter that signals that. So by manipulating then the gut biome, or the bacteria that lives in the gut, you are then becoming, your immune system is then decreased. Also, obviously, your ability to absorb nutrients to repair your body would be decreased as well. And then also all the different hormones and neurotransmitters that signals all the different activities throughout the whole body, all of that is impacted as well.
Correct. The GI tract is referred to now as our eighth organ. You have 10 times more cells in your GI tract than you have in your entire body. So that that's really a very powerful machine. And Shihata and Kruger and Stanley, a number of scientists, in studying that gut microbiome, have shown how critical those organisms are. And it's interesting that with the fecal transplant, how many of those diseases can actually be cured if you get the right mix of organisms in that transplant that you're inserting. Now, if you don't, it can also have some negative impact because it works both ways. But those organisms are critical, but all of the beneficial organisms are all very sensitive to this powerful antibiotic that we call Roundup or glyphosate that Monsanto had actually patented as an antibiotic. But again, it's an antibiotic against the good guys and not the bad ones.
So, and good bacteria, in addition to everything else that it does in the digestive process, it is also crucial then in the production of a number of vitamins that we need just for metabolic activity, for cell function. And we know that every disease pretty much starts in the cell. If there's a dysfunction within the cell, then the body cannot thrive. And so by then reducing these beneficial bacteria, killing them off with glyphosate, you're then also eliminating your ability to support your own cell function, which means that you will not be able to withstand disease as well, and you'll die sooner.
Right. You essentially become malnourished in spite of what you're eating because you can't absorb it. It's not in the right form or the right combination, so that your body can't utilize that material then for cellular function. And the glyphosate is accomplishing that in two ways. One is by its antibiotic activity against those organisms, but then also serving as a very powerful chelator. A chelator is just a chemical that can grab onto another element and immobilize it so that it's not functional. So that by tying up those minerals, all of our enzymes are essentially metalloproteins. Well, the metallo part is there as the cofactor, again, for function of that protein. And when you tie it up or chelate it so that it's not functional in as a cofactor, then you've essentially destroyed that enzyme that we depend on so critically for our overall cellular function.
So in essence, we pretty much, with the GMO, we created a worldwide experiment that we have no idea what the end is going to look like. And there's been no testing prior to implementing this technology, of my understanding. We're just, the experiment is us right now.
It's definitely an experiment. Dr. Lauren Pang, who is health director in Hawaii, was on a panel that I was on about a month ago in Boston, and he has suggested that we reactivate the Nuremberg trial because he said this is an experiment that no one has given consent for. He said it violates all of our consent clauses, and you can't say that, well, if you don't agree to this, that you'll go hungry. That's under duress. You can't get common consent by threat or by duress. But he said there's no experimental information to go on. He said when he would, as an MD, would write a prescription for a patient, he would say, now these are the side effects, and you may watch for them very closely. We can't even do that with the GMOs because we don't know what the side effects are. We haven't studied the chemicals that are being produced, that we're engineering those to either produce or to tolerate in a safety manner either, that we can answer any of those questions. So you can't ask your doctor and say, well, if this doesn't work, is there an alternative? If I go off the medicine, does that solve that side effect problem? We can't even answer that. And so Dr. Pang had suggested that the enormity of the problem, the health factor and disintegration of health in the United States but worldwide, is reaching the point where the Nuremberg trials for medical experimentation and that should be reactivated. Certainly, if you look at the CDC data for 32 diseases, and you mentioned a number of those from cancer on, if you look at the CDC data, just the epidemiological plots have a 0.9 to 0.99 Pearson's correlation coefficient with cause-effect relationship. Now, correlation doesn't always mean cause-effect, but when you get 32 diseases that all follow the same pattern and you can connect all of the biochemical dots back to glyphosate or back to the GMO alterations, then you have something that should be a red flag that is mandating the scientific studies, and yet they're still not being done. They're still prohibited by the companies, and you're threatened with your job and everything else if you would even be able to find the funding to initiate those studies. But if you look at the epidemiological curves and realize that medical data is 8 to 10 years lag before it really gets into those curves because of the reporting process, that you can, if you project those 8 to 10 years, you can literally see that we're on the brink of the world's largest depopulation that has ever been recorded in the history of this earth.
That's just horrifying. We'll be back in just a little bit with Dr. Don Huber talking about GMO and the impact of GMO on us and worldwide. Welcome back to HealthMade Radio. I'm here with Dr. Don Huber. We're talking about the implications of GMO foods. GMO was something that was supposed to help us to solve world hunger, and now we are dealing with a health crisis because of that. So you're talking about that the investigation in regards to GMO, it's very hard to do. And there's a lot of financial pressure, obviously, to stop these type of investigations. Why is that so?
There's a lot of money involved in it. When you can patent a basic life process, such as a seed and a crop, you literally have tremendous control and a monopoly on that particular entity and society as an essential entity. So that if you could patent water, you could dictate what the cost of that was going to be, and that's what happens with seed. That the cost has increased 15 to as many as 500-fold from what it was before some of those seeds were genetically engineered. Problem is you can't get them unless they're genetically engineered, because all the seed companies have been purchased by those companies, so that there's a monopoly on them now and a control that goes with it. And with that control then goes the money that brings in, for one company, about $8 billion a year just in royalties on their patent. They're not having to do much work for that. They still get all of the income from the seed that they would normally get, but it's the tech fee that then gives them that extra slush fund to buy politicians or run lawsuits and a number of other things that are done there, or to buy up more companies to increase that power and control that they have.
So you would think that we have antitrust laws, to protect us from such monopolies. They are not in effect in regards to this issue?
Well, apparently not. We just saw a $66 billion merger approved by the antitrust organization, the Department of Justice, whoever controls that in the States and also in Europe, with the merger of Bayer and Monsanto, or the buyout of Monsanto by Bayer. So that apparently not much concern there. They did have them divest themselves of $9 billion, but when you're spending $66 billion and also have that kind of a control on the commodity, that's an essential commodity, it doesn't make much of an impact. It's kind of like that $19 million fine that EPA finally got through the courts for fraudulent safety testing here in 1991. It was just slapping the hand when you consider the $9 billion or the $8 billion income from that process, and so not much changes. Each entity has to go through its own lawsuit to challenge, and start with, you don't have access to the materials to do the research. And the other thing, if a young scientist wanted to do what I was able to do in my last 10 or 15 years of active career, in 3 or 4 years, they'd find they were looking for a job. There's too much money, too many endowments, and too much control and censorship involved in academia as well as in industry.
So in essence, if you would want to investigate, you, A, would not have the money to do so. 2, these companies obviously can control and put a lot of political pressure, with the money that they have. Then 3, as a scientist, you can pretty much say that your career is over, and whatever you worked for, your whole schooling, your whole life, you will lose that because you will not be able to practice science anymore.
That's correct. And you can add another aspect of that is that you can't get access to the product. Yeah. It's a proprietary product, patented, and unless the company tells you that you can use it, you're in violation of the patent to even consider the research on it.
So you wouldn't even be able to study the product to see the harmful effect that it's had.
That's correct, and that's been enforced in the courts by the companies. For a farmer to even purchase the seed, and he's not purchasing the seed, he's leasing it. If you read the technology agreement, he's just leasing that seed from them, but he assumes all liability for any damage that may result from it. He waives all damage to the companies, but he has to agree not to do any research, not to do any comparisons, and to purchase the company's product that it's engineered to tolerate. So they catch him in a number of different ways in that process, and what you see then is a servitude starting or being built up to where a farmer is in India, for instance. They get into debt so far, or when they have a bad year, they can't pay for the seed, and you have 2,000 suicides a month in India because of that indebtedness. In order to relieve the debt from the family, the farmer will commit suicide. So you have a number of different things that come into play there with the patent and with the censorship and power and control that they have over these processes.
Seems like an amazing business model to lease the seed, and so the farmer does not even own it. In addition to that, you accept no responsibility in any shape or form for the product that you're supplying.
Right.
Yeah, that is fascinating. Well, this discussion about GMO, I think it's such an important discussion, and it's something that impacts us in so many different ways. So we are actually going to have another show on it where we want to talk a little bit more about how we can go about solving this crisis that we are in the middle of right now. And I will be talking with Dr. Don Huber next week. Please tune in. This is vital information. That is all for today. I'm Dr. Michael Karlfeldt with HealthMade Radio. Connect with us at healthmade.co. Health is what you make it. And tune in for next week.